In this presentation, I shall outline some key principles in the regulation of plasma products through the oversight of their safety, quality, and efficacy. A shortened version of this presentation was given remotely to the Congress of the Asian Association for Transfusion Medicine in Mumbai on the 15th November 2025. The World Health Assembly has recognized the importance of regulatory control of all blood products. In its Resolution 63.12, the Assembly emphasized the need for strong regulatory authorities nationally and regionally as a fundamental requirement to assure global availability of these important therapies. It is customary, particularly for the regulated sector, to refer to the regulatory principles of safety, quality, and efficacy as hurdles to be overcome in order to assure access to blood and plasma products. I do not favor this designation myself, as it implies a negative connotation for these fundamental principles. I want to point out that the main hurdle to be overcome to assure access is not regulatory but financial, as any products approved by regulators have to be paid for if they are to be accessible to patients. This fourth hurdle, however, is beyond the scope of this presentation. For plasma products, the most significant contributors to these principles are shown. In relation to quality, we think mostly in terms of good manufacturing practices, which are referred to as GMPs. The most important aspect of safety, although certainly not the only one, is safety from the transmission of pathogens. And we recognize the importance of clinical trials in assuring efficacy. GMPs are a set of practices which are universally applicable to any process of manufacture, including plasma products. The principles are shown and include written procedures for step-by-step stages in manufacture and written instructions defining workplace standards. These procedures need to be followed meticulously, without modifications or shortcuts. All work has to be documented for compliance and trackability, and a chronicle of the process and procedures needs to be completed. Systems and processes need to be validated to ensure that they are working as designed. Again, this validation process needs to be documented to ensure quality and consistency. The equipment and the facility need to be adequately maintained to minimize quality and safety concerns. This also needs to be documented. A well laid out plan to ensure the productivity and quality of facilities and equipment needs to be designed and implemented. Quality must be built into the product by controlling all aspects of the manufacturing, including packaging, labeling, and distribution. Employees need to be competent in their roles through defined and demonstrable criteria for competency. Contamination must be avoided through developing and defining standards which are implemented throughout the workplace. Finally, the continued implementation of these principles needs to be followed and demonstrated through regular audits to validate compliance. Safety from the transmission of viruses through plasma product use was assured through the implementation of viral activation processes embedded in the manufacture, and this has only been achieved for the past 30 years, in the wake of the catastrophic epidemics in the hemophilia population in particular over the 1970s to the 1990s. At the same time, the selection of safe donors and screening donations for viral markers has increased greatly the transfusion of blood bank components, but small transmissions of viral agents, including emerging viruses, has continued to occur in the transfused population. The concept of a safety tripod has been developed to emphasize the three pillars of blood safety: selection of donors which are safe, testing donations for viral agents, and reducing any pathogen contamination through reduction processes in the manufacture. As stated, for blood components, the third pillar of reduction has not been fully and universally implemented. In contrast, for plasma products, viral reduction steps integrated into the manufacturing process are mandatory for all products and form part of regulatory requirements. Hence, the safety of plasma products is strongly dependent on these processes, and we think of plasma product safety as resting strongly on many pillars of pathogen reduction. I will now turn to some aspects of documentation which are available publicly to those interested in how regulatory control is exerted globally by the main regulatory agencies. An abundance of documentation is available, and I do not think that many of you have the time and inclination to review all of them, although they should be studied by all those working in regulatory authorities. To access the principles used by the United States Food and Drug Administration, this paper, available freely on the internet, provides a useful synthesis. The author is Dr. Mark Weinstein, who is a former senior official in the FDA's Office of Blood Research and Review and was charged with overseeing plasma products for many years. These extracts from Dr. Weinstein's paper summarize several aspects of the FDA's framework, and I show them fully aware that it is not possible for you to assimilate these through simply scrutinizing the slide, however long I put it up. It emphasizes the breadth and complexity of the FDA's framework for oversight, including how the GMPs and the viral inactivation processes are regulated. And going to Europe and assessing the processes implemented by the European Medicines Agency and similar bodies, a plethora of regulations, guidelines, and standards are in place, some of which are shown on this slide. Again, this just shows the complexity of the framework and should engage all of you who are working actively in regulation yourselves. But for those interested in a synthesis of all these measures which is more accessible, I refer you to these two works which are available on the internet, which summarize and exemplify the principles in a simpler fashion. These deal with the issues around two main classes of plasma products, which include coagulation factors and immunoglobulins, but the principles are applicable to all the products. I will now outline the principles described in these documents. The regulatory authority must have full information on the plasma used as a raw material for the manufacture. These requirements include ascertaining the regulatory status of the plasma supplier, establishing the donor epidemiology, establishing donor exclusion criteria to ensure recruitment from a safe population of donors, stabilizing the screening tests done on the blood and the plasma, any quality assurance measures, description of any inventory hold used to quarantine the plasma before it is released for manufacture, delineation and specification of the size of the plasma manufacturing pool, also its testing. A very useful European document which includes all this information which must be submitted by the manufacturer to the authority is the Plasma Master File. This is compiled by the manufacturer in collaboration with the suppliers of the plasma, and it should include all the aspects of plasma raw material safety as shown. This document should be studied and its requirements implemented by all regulators, irrespective of where they exert their authority. It is noteworthy that, Because of the global nature of the plasma industry, most of the plasma collected in the high-income economies is characterized through such a plasma master file. The regulator must have full assurance of the manufacturing process through gathering information and assessing it on the crucial manufacturing steps and related in-process controls, the viral inactivation and removal steps, process consistency, and the batch release specification. I will now give some examples of crucial aspects of the manufacturing process. This is an extract from the guides for plasma product assessment which I showed earlier. I emphasize that these documents are freely available through searching on the internet. It outlines the information required from a manufacturer regarding the manufacturing process. These include the license issued by the regulatory agency in the country where the manufacturing facility is located. Also required is an outline of the critical steps of the manufacturing process, as shown in a flowchart. The crucial information regarding viral safety is also required in the form of validated studies which show the elimination of the relevant transmissible viruses or models for their quantification, as listed in the slide. This should then be followed with an estimate of the residual risk of transmission for the main relevant viruses, HIV, HBV, and HCV. Detailed descriptions of these calculations are available on the documents cited. Coming again to the crucial area of viral safety, we need to keep in mind the nature of the process in the sense of commencing with the pooling of thousands of donations, which is necessary for GMPs in producing batches of product which are consistent and characterizable. However, this imposes the strong need to ensure the exclusion of any contaminated units in order to minimize exposure to pathogens by patients. Each step in the manufacture has to be defined and in-process controls assigned to it, which enable its conformity to GMPs to be assessed and assured. In this process for an immunoglobulin product, these controls are specified for the various steps, and I draw particular attention in this particular manufacturing of an immunoglobulin product to the 3 steps claimed by the manufacturer to contribute to the viral safety. This example is extracted from the literature and is also available for public comment. Here we see some manufacturing process flows for the intravenous immunoglobulins currently available on the market with the viral inactivation steps identified. There would be other viral inactivation steps, all of which require validation. Process consistency needs to be demonstrated by measurement of in-process controls previously identified as relevant describers for the characterization of the process. A batch release specification needs to be developed and adhered to for every batch released. It should be based on the pivotal characteristics of the product as specified in documents such as the relevant pharmacopoeias. The batch release specification is built up from the detailed characterization of the product, with each measurement justified and validated against the product's intended use and fitness of purpose. In this slide, we see this for a recently approved immunoglobulin product, and the reference is available for you to access the relevant publication. Finally, the product itself, formulated and dispensed, needs to be assessed by the regulator for the potency of the product and shelf life, other markets where the product is available, its history in these markets, and clinical studies demonstrating the product's efficacy. I will show some examples. The stability of the product over its claimed shelf life needs always to be assessed for all the conditions which the manufacturer wishes to use. In this example, a particular albumin solution stored at different temperatures is monitored over storage for levels of the contaminant prekallikrein activator, which has been implicated in producing hypotensive reactions in patients given albumin products. It was observed that pKa levels rose unexpectedly over storage at the temperatures envisaged for the product's storage. This was subsequently investigated and resolved. It emphasizes that nothing can be taken for granted and that stability studies are needed to support the requirements of the shelf life. Here we see stability studies for relevant parameters in immunoglobulin products. These include the maintenance of the biological function of the Fc portion of the IgG molecule and the generation and presence of dimers and aggregates, which are of importance in avoiding reactions in patients. The regulator should request information regarding the product's approval status in other markets. Clinical studies and any adverse events recorded are mandatory for ensuring efficacy and safety. Clinical studies are essential. I never cease to reiterate this. Let us look at this trial, which was proposed in 2010 and subsequently carried out and published, known as the CPET study. In this trial, patients who were previously unexposed to products in hemophilia were randomized to first treatment with plasma-derived or recombinant products. The plasma-derived products were factor VIII von Willebrand factor concentrates, while the recombinant products were the first-generation products generated from rodent cell lines, which lacked von Willebrand factor. The study was carried out over the early years of the 2010s and published in 2016. The results indicated a significantly higher incidence in inhibitors to factor VIII in patients given only recombinant products. This study's publication in the New England Journal of Medicine has renewed therapeutic interest in the use of plasma-derived factor VIII. In these studies shown on the slide, on the pharmacokinetics of immunoglobulin products, we see the variations in immunoglobulin levels post-administration for products manufactured in different ways and administered through different routes. Early preparations of intravenous immunoglobulin treated with enzymes to remove dangerous aggregates showed lower half-lives compared to other products. Modern preparations have avoided these problems. With subcutaneously administered immunoglobulin, a steady state of IgG levels is the objective of the administration to result in a protective level for several weeks. The capacity of the products in mirroring this desirable profile needs always to be assured. A number of large pragmatic trials have assessed the role of albumin in sepsis in the past 15 years. The SAFE study in Australasia, the EARS study in France, and the Albios study in Italy have all studied this therapy. Interestingly, the trials consistently showed a beneficial effect of albumin infusion on survival, and the pre-specified subgroup in the SAFE trial did show a significant effect. However, albumin infusion did not show a significant effect in the overall trials. In the Albios study, performed on severely septic patients in Italian centers and funded by the Italian Pharmaceutical Agency, a post-hoc analysis showed a beneficial effect in septic shock, which achieved significance. The post-hoc nature of this analysis deflected its importance, although it has to be said that the relevant journal, the New England Journal, was excessively purist in its refusal to allow this result to be included in the paper's conclusions. I find this representation of the progression of regulatory oversight in all transfusion medicine to be a useful illustration of the balance that can be achieved between the relaxed attitude of regulators in previous eras and the rigorous frameworks developed partly as a reaction to the mistakes which ensued from this attitude. The pendulum should fall at the bottom of its arc, and the ultimate aim is the designation of an appropriate risk-to-benefit ratio for the product. This principle is described in more detail in a publication called, as is the title of this slide, The Regulatory Pendulum in Transfusion Medicine, which is available online. Before I come to my final reflections, I wish to reiterate the essential role of regulation in assuring the safety, quality, and efficacy of plasma and blood products, in protecting patients from infection and ensuring their well-being. I was appalled at noting this recent incident in India, and I would just comment that, in the presence of the measures for assuring safe donor recruitment and the screening of donations with adequate tests, measures which should be imposed by regulators, this tragic incident and others like it would not have occurred. And so I conclude with these final reflections. The plasma product sector is heavily regulated, and quite rightly so. Because of this regulation, plasma-derived medicines manufactured in the jurisdictions of the main regulatory agencies are very safe. The problems arising from the past, particularly in the viral safety area, have contributed to this. We need to recognize historical events and learn from them if we are not to repeat them. The basis of regulation is the precautionary principle, which must be applied firmly but judiciously. Using this principle, any uncertainty in the safety of products is resolved entirely to the benefit of the patient, if necessarily through restricting plasma supply or product release. The principle also requires constant review of such precautionary measures and revision when it is justified. We have seen this in recent years with the issue of donor deferral for people who have lived in the UK through the period of risk for transmission of BSC and VCJD. Investment in regulatory provision will dissuade the responsible individuals from attempting actions such as counterfeiting, making false claims, etc. And these individuals exist in all countries where I have been a participant in this industry. All countries, I assure you. Harmonization of regulatory measures is desirable to alleviate the burden both on industry and the regulators. The principles outlined in this presentation are all embedded in the requirements of the International Conference on Harmonization, and these are also publicly available. And this brings me to the end of this presentation.